what was the inspiration to do this review series on fat-soluble vitamins? You know, why is this timely right now?
Well, clearly this represents, 2013 represents the 100th anniversary since the first description of what is accepted to be the first description of vitamin A actions in the literature, published in the JBC. And I think, you know, this was 100 years ago. There was an article published by McCullum and Davis, and shortly thereafter there was one published by Mendel and Osborne, both of which involve studies carried out in growing rats, where the diet of the rat was the fat, basically content of the diet, put into the fat that was put into the diet was basically extracted with ether, and that removed some materials which proved to be essential for growth of the rats. Subsequently, that ether extractable material became known as fat-soluble A, and fat-soluble A evolved into vitamin A. So this marks the 100th anniversary of those two publications in 1913, which are basically accepted as the initial molecular description of a vitamin, and not specifically of vitamin A.
In the body, what are the physiological processes that are affected by vitamin A?
Well, there's obviously the classical one that I learned over 50 years ago when I was in elementary school, was vitamin A is needed for vision. And basically, when I was in elementary school, that was really the only function of vitamin A that was well established. And George Wald and many others were very seminal in carrying out biochemical studies of the actions of vitamin A in vision. When I entered research, basically as I ended my graduate career, and I went into studying vitamin A and how it is metabolized and how it acts in the body. This is more than 30 years ago. It was beyond understanding how vitamin A acted in vision. The actions of vitamin A outside of vision were not well understood at the molecular level. It was clear that vitamin A is not only needed for vision, it's also needed to maintain normal growth and development of an individual, whether it be a human or another mammal. Vitamin A is required for growth and development of basically from the embryo through the adult. And basically that's because vitamin A affects very prominently cellular differentiation and cellular proliferation. So growth and development is one, and sort of as a subset, vitamin A is really important for maintaining a normal immunity. It's important for both spermatogenesis to take place, also for oogenesis to take place. It's important for maintaining epithelial linings such as barriers like the skin. So vitamin A acts in many, many different organs in the body. And it's not really associated with just, even though we might say vitamin A and vision, it's associated with so many other things. It's required for so many other things in the body. So,
what kind of diseases result from vitamin A deficiency? Obviously, that's going to have a big impact on vision, but is there anything else that really is of major concern and sort of the driving force behind a lot of the research of vitamin A?
Clearly, vitamin A deficiency remains a major public health problem throughout the third world. You know, World Health Organization estimates are, you know, a staggering number of children, 6 months to maybe 5 years of age, who are at risk of vitamin A deficiency. The classical symptom of vitamin A deficiency, the first symptom is night blindness, progressing into xerophthalmia. Ultimately, if vitamin A is not given to the child will die, usually because of infectious diseases. The immune system is impaired. Not only does vitamin A administration to these at-risk children prevent mortality, but there's a large literature now that suggests vitamin A interventions to at-risk populations of children also has an effect on the morbidity of those children. That is, they have fewer colds, they have fewer days lost because of illness. So vitamin A clearly is one of the big micronutrient deficiencies across the planet. Let me also say that not only is too little vitamin A, bad. But in our society, in the first world, basically in the developed world, um, we're starting to encounter a lot of subclinical vitamin A toxicity. So vitamin A is also can also create problems at the other end of the intake spectrum. Too much is also bad.
So this review series that we just ran in JLR has 4 different articles. So how did you pick those 4 different areas? I mean, what was the rationale behind that?
Well, I guess it sort of reflects my view of the work that's going on in the vitamin A field. And I have to say, because we were doing not only vitamin A, we were doing vitamin E, we were doing vitamin D and vitamin K, I tried to choose a number of articles for each of those vitamins that reflected the research interest, the research activity within those different areas, within those specific vitamins and the research that's ongoing. So I selected a paper by Earl Harrison and his colleagues, or asked Earl Harrison and his colleagues to provide a review focused on carotenoid metabolism and specifically on a reporting on apocarotinase. So what you have are some vitamin A metabolites, and up top you have the structure of all-trans beta-carotene, and then you also have the structure of vitamin A, which in the red, underlined in the red. And it's pretty clear that if you cleave the central 15-15 prime double bond of all-trans beta-carotene, so if you just look right in the center of that all-trans beta-carotene molecule, there's a double bond. So if you look at the structure of all-trans retinol, it's real obvious how they're related. That central 15-15 prime double bond is cleaved by an enzyme known as carotene monooxygenase, forming retinaldehyde, which is then reduced to all-trans retinol. The all-trans retinol could either be esterified to retinol ester, which I mentioned earlier, or it can be oxidized. It can be oxidized to retinoic acid, which is for either all-trans or 9-cis-retinoic acid, which are the ligands for the transcription factors, the RARs and the RXRs. So what Harrison showed was it's real clear that central cleavage, about the 15-15 prime double bond, that central double bond, could basically, that's the source of retinol. And that's been known, not just Harrison, that's been known for the last 15 years, that carotene cleaving, carotene 15-15 prime monooxygenase 1 was the enzyme responsible for that. But there's also another enzyme, the so-called eccentric or asymmetric cleavage enzyme. Carotene, it goes by CMO2 or CDO2. There's a little bit of controversy whether this enzyme, the second enzyme, is a monooxygenase or a dioxygenase. But the second enzyme cleaves asymmetrically. That is, it cleaves about other of the double bonds present in beta-carotene. And when that happens, what you get is a molecule which is a little longer than retinol, contains extra carbons, and you get a molecule which is a little shorter, contains fewer carbons. And those eccentric cleavage products are referred to as apocarotinels. The apocarotinels, just like the retinols, can be either reduced or oxidized and can be esterified. So there's what Harrison showed, and this is to me a major finding in the last couple of years, what Harrison showed were some of the apocarotenoids that were produced via eccentric cleavage of beta-carotene basically were potent antagonists of RAR signaling. What that means is that they potently block retinoic acid activation of transcription. So that means they potentially have a modulatory role in regulating vitamin A action. And I personally find that to be a very exciting finding, something that was published in the JBC last year, 2012. And it has potential ramifications, big ramifications potentially, for understanding how vitamin A acts and what it does in the body, because these eccentric cleavage products are potentially modulators of vitamin A action. So I wanted to have a chapter on that.
What do you see are some of the grand challenges in the next 5 years in vitamin A research?
Well, I guess at the molecular level, understanding the non-genomic of actions of vitamin A is perhaps fundamentally important to understanding what vitamin A does. And this leads into a number of questions about relationships between vitamin A action, vitamin A metabolism, and metabolic diseases. So one of the most exciting areas out there, and very controversial, is a literature, very substantial literature, but I need to say also there's a substantial literature that argues against this idea, and the idea is that retinol binding protein, or RBP, is synthesized and secreted by adipocytes, and that this adipocyte, this adipokine RBP, then somehow through some people are proposing mechanisms, but in some way affects or impairs insulin signaling in the body, giving rise to type 2 diabetes. And so that potentially RBP may be a link between development of obesity and type 2 diabetes. So it's potentially a very important molecular link. This is an argument put forward by Dr. Barbara Kahn. And also let me say that I'm using the acronym RBP. The genetic nomenclature for RBP, RBP standing for retinol binding protein, the genetic nomenclature, this protein, or the gene that encodes this protein, is RBP4. So sometimes, or actually a lot now in the literature, you'll see this protein described as RBP4, and the association between RBP4 and impaired insulin responsiveness, and then that's been extended into development of fatty liver and liver disease, also vessel wall disease. So for me, understanding how, if, why these metabolic disease relationships with RBP, whether they involve vitamin A, whether they don't involve vitamin A, whether they involve non-genomic actions of vitamin A and vitamin A related proteins, that is really an area over the next 10 or so years that will probably play its way out. So for me, non-genomic signaling and how this might link into development or prevention of metabolic diseases like diabetes, like obesity, like cardiovascular disease are very, very topical right now. So for me, that's the major, that's the future direction, understanding those at the molecular level.